Gardunia. Unfortunately, our original speaker, John Hickey, was unable to make it, but thankfully Brian came right at the end to make sure that we still had a speaker. So we're very grateful to have him. Brian completed his bachelor's in plant genetics and breeding and his master's in agronomy from Brigham Young University. He then went on to complete his PhD in plant breeding from Texas A&M University. He began his career as a popcorn breeder with Ag Alumni Seed for 3 years before joining Monsanto in 2009. He is currently working at Bayer Crop Science, where he's the head of cotton product design. He leads the team responsible for population improvement, parent selection, genomic prediction, and inbreed selection. One of his other main current responsibilities is as the technical project lead for collaboration with Abacus Bio to develop bioeconomic selection indices for multiple crops and regions. And I'll pass it over to you, Brian.
Okay, and I have some intro stuff here too. So, and for those, I don't know how many, like how your normal kind of culture is for these, but I have no problem if people want to jump in and interrupt me. I may struggle to see the chat. So if people are asking questions, I'll try to have that up on another screen. If that is something that you guys use, let me know if I need to be watching that. But hey, Mike.
We usually keep the questions to the end, but if people do want to throw them in the chat, I can keep an eye on it, Brian, and let you know.
So yeah, at least here at Bayer and at Monsanto too, being interrupted is kind of the state of the state. So I don't mind at all if you guys, if you got something you want to jump in and talk about, or questions as we go, it won't bother me at all. I don't have to get through the slides. I have no objective other than to have a conversation with you guys. So that's what I'm here for. So I did have kind of a little bit of an introduction slide to myself here. I've moved mostly all around the Midwest, working on plant breeding and genetics. This was my backup career. I wanted to be a violinist at one point in my life. And then I found being a professional violinist was really hard. And maybe wasn't good enough of a violinist to pull that off. Was in Nicaragua at one point as a Mormon missionary and got really interested in genetic, in ag. So I was on a bus one day near the end of my time there, and I was talking to this guy on the bus about rice, and he grew a short grain rice, but he wanted to grow long grain rice because long grain rice is what they eat in Nicaragua, and he exported to the US and imported from the US. And he thought this was a silly state of affairs. So he's like, why can't we figure out the genetics to get a high of rice variety that's the type of rice that I want that'll do well here in Nicaragua. So I had this in my head when I went back to school. I took a genetics class and just fell in love with it and had a lot of really lucky opportunities where I was able to go work in Florida on tomatoes, and I was able to get a job working in a molecular marker lab, and I worked on all the old marker types. So that was my expertise in graduate school, as I started with Southern blots and worked my way through rapids and SSRs and AFLPs and up to SNPs by the end, but like all the different types of making and using markers. I worked on Chenopodium quinoa, developing mapping populations. This is me on the bottom there making crosses down in Bolivia. Those were all the ones with the little bags on them were my populations I was making. And then I worked on cotton at Texas A&M with Cotton Incorporated, and then was very burnt out of school and left to go to be a popcorn breeder, mostly focusing on popcorn for South America. So I was in Indiana, but I spent most of my time thinking about hybrids for Brazil and Argentina. And then went to Monsanto as a discovery breeder, where I knew Mike Loheis, who I saw was on the call. and then moved to St. Louis, where I've led different kinds of teams. And I wish my job was me out in the field all the time now. Unfortunately, a lot of my job is more like this bottom picture where pre-COVID days, there were a lot of people. And then for a while, I was working out of my basement. So a lot more time in front of a computer and with people than not. This is my team, or most of my team. I have a couple of people that came that weren't in this field tour, but we work. I'll just try to describe as we go through here the corn, like the breeding process somewhat, and why our teams are divided up, why we are. But essentially, the way you can think of a plant breeding program is a series of funnels where we have a bunch of inbreds at the top of that funnel, and we're selecting down to the few inbreds that we're going to commercialize. And we've divided up our teams in Bayer into what we're calling a design team and a development team. Essentially, that's different parts of that funnel, and my team is at the front of that. So the parent selection, population improvement, genomic prediction, and then those first selections in the field or before they get to a yield trial. And so let me essentially what the story I want to talk through is some of the reasons why I think plant breeding and animal breeding have diverged and are kind of different from each other, and how they come back together. And this was my hook as I thought of what to talk about. I talked about this at Rosalind Institute this summer as well. And this kind of started me thinking about this, because there's probably an alternate reality somewhere where I'm an animal breeder instead of a plant breeder. I mean, maybe I took a job, could have taken a job, but from my current PhD work, which was all plants, but I really loved animal breeding actually. And my undergrad advisor was an animal breeding advisor, and he worked on sheep. And the point of divergence for me was he had invited me to go to the field, go to the farm and help emasculate sheep. And I did not enjoy emasculating sheep. So I went to plants. It was a much cleaner emasculation process. And but I had a little hobby farm. So this is the alternate me. This is my pig that I raised. These are my kids and in both these pictures. But one of the things that I tried to do at one point in my career here when I was at Monsanto and at Bayer was to try to think differently about plant breeding programs and how to envision plant breeding programs in a different way. And one of the ways to do that was to think, well, what would an animal breeder do? How would this look differently if it was an animal breeding program? And that's part of why I started collaborating with John Hickey, with Gregor Gorjanc, with some of the animal breeding folks, and why I can still have a collaboration with Abacus Bio is trying to think, what are the ways that plant breeding is different or the same as animal breeding, and what do we pull from those different fields to help improve how we do breeding at Bayer? So in doing that, I have this hypothesis, and this is Brian's brain. This is not Bayer policy. This is not the company line, right? So when I look at the history of plant breeding, and I think about the points where the different breeding groups diverge, and I think there was a divergence. Like I went to Texas A&M from 2002 to 2006. One of my colleagues here at Bayer is Edward Cargill. He was at Texas A&M in 2002 to 2005 and was a PhD student in animal breeding. My field was across the street from his field. So his field, where he had fistulated cows, And then there was a dog breeding program that also had like dog kennel runs in that field, was across the street from where I had my cotton fields. I never met them. I had no interaction with anyone from the animal breeding department that was of similar size and quite high quality of science that was going on there. And so as I think about why the training, we didn't take classes hardly together even, like the base genetics classes, maybe stats classes, the cytogenetics class was taught together between animal and plant breeding. But other than that, we didn't take the same classes. And this is my hypothesis on why there's been kind of a divergence and why there's a difference in mindset and a difference in focus and methods between plant and animal breeding. I think there's kind of three interacting factors that have led to this. Some of those factors may have some reasons why there's more of a convergence of ideas and methods, I think, today than there was in 2006 between plant and animal breeding programs. And I think there's a lot of reasons why we should be working together more to develop methods and to think critically about how we do breeding, because some of the things that made plant and animal breeding different, I think, are starting to make them more the same. And that's some of what the conversation I wanted to have with you guys today. So those three areas is, first, there's a biology area. So plants, the thing that made plants, that made the biggest advance, I think, for plant breeding was maybe not even understanding of genetics, but the ability to make inbreds and hybrid clones easily and cost efficiently that we could use for both breeding evaluation and for sales. And I'll explain why I think that was such a key important thing and how that shapes how an animal breeder makes decisions and shapes their breeding program. The next part of that is the business. There's a business side of this that where, and maybe my bias is for that is I work for a big business that does breeding and genetics, but there is a seed industry that grew from having improved inbreds and hybrids. So these clones became themselves a commodity that could be sold because they showed increased yield and adaptation to modern agricultural systems. So there was co-development of a system of agriculture and the business of the seed industry. And this is where you see graphs like this often in corn hybrid breeding slides where they show open pollinated, double cross hybrids, single cross hybrids, where really the reason why those things are being graphed that way is not just to show the biology of hybrids, the types of hybrids that were used, but it actually shows how the business is tied directly into that biology system. And then to support that business is really relies on an intellectual property system and political support that enables both the breeding, the producer, and the farmer to be successful in this kind of system. And it supports the biology that plant breeding is using in releasing and selling, and it supports the business models of the companies and even the public institutions that are breeding for plant breeding. So why is this? You know, here's a picture on the left of a corn plant. The biology of a corn plant that really matters for breeding that makes corn such a great hybrid system is you've got anthers on the top, that's your male pollen source. You've got ears on the bottom, that's your female seed source that where all the eggs are. It's really easy to chop off the top of this tassel and move that pollen to another place and drop that pollen on a different set of ears. So, and to make an inbred that is pretty straightforward as well. You just put a bag over the ear and you put a bag over the tassel. so that they're, you don't get pollen that you don't want, and then you switch those bags when you want to self-pollinate that plant, so then you have a self-pollinated ear where all the seed come from a controlled pollen source. And the other thing you get is, these are ears from a paper from about Barbara McClintock, but the thing to notice is, you're getting 200 seed or more from each pollination. Soy and other things have different biology that I won't go into, but that we manipulate other ways. But essentially, this makes it really easy to increase the seed, and each seed is a plant. So if we're going to plant 30,000 seeds in a field, you can count backwards to how many ears you need to make of the hybrid, and you can then figure out how many plants you need to plant to make the hybrid seed, and cost that out of how much it's going to cost you to produce that clonal hybrid to sell as many acres as you want. And if so this biology component is what is really a key thing for plant breeding. The traits that we're selecting for, we have kind of two levels of traits. One is the traits that are economically important for the grower. Yield of seed, if that's what they're selling, or biomass, if they're using the whole plant and feeding that to cows, for example. But also the things that make it easy to produce the hybrid. So it's inbred yields, it's amount of pollen that they make, and how easy it is to detassel, and the size of those seeds, so it's easy to package and sell to a farmer. And in that, you can see the way that we're thinking about it. The other thing that the bio, and I'll show this in the next couple of slides, is with this biology, unlike a herd of cows, you're able to control completely the genetics of every plant that you put out in the field. And you could plant a field of heterogeneous genetics, but you could also plant a clone that's exactly the same for every plant in that field, and it's the exact same process. So on the other side of this slide, I've got from a paper by John Hickey that was in Nature Genetics that I'll talk about again at the end. Is he is arguing that in these papers that there's an alignment of the biology and the methodology that starts to bring plant and animal breeding programs and genetics and breeding closer together. And he's got here the different things that are inventions that are important for plants and animals. And I think one of the ways that this biology starts to be, if you look at the important inventions for plants, a lot of these actually are around understanding this ability to make clonal hybrids and inbreds and reproduce them. You get single seed descent breeding, you've got pure line breeding theory, which is you're going to self that until it's an inbred, fully inbred. You got heterosis there, you're making a hybrid, modern pedigree selection, and a lot of this that heads through. The key thing that I'm going to come back to, that if you compare these lists, that is really different, is if you look on the analysis side, the genetics advances are important for plant breeding, but they're especially important for animal breeding, because you don't have this ability to precisely control the flow of genetics and to create clonal hybrids or clonal inbreds in the same way. And so you have an increased focus on breeding values and random effects and genetics and models to be able to predict performance of individuals. Plus, you're not able to replicate the phenotypes, and I'll get to why that changes the way you think about the structure of your breeding program a lot. But if you look at when breeding values as random effects becomes really important on this list for plants and animal breeding, it's on this list here in 1950 for animal breeding, and it's on this list here in 1998 for plant breeding. And there were plant breeders who were using breeding values as random effects. But I'll tell you, at the popcorn company I was at, which is, you know, a highly profitable little breeding program, we weren't using bluff. We just were using the mean performance of our inbreds, and that was in 2009. So so we'll come back to this, but this biology thing is really important to get your head around why plant and animal breeding programs are different. And one of the ways that I think you start to see that is if you think about the way you would make an experimental design for measuring the selection of traits and the selection of individuals, because your objective is to create a clonal hybrid or variety at the end that you're going to sell across a lot of acres. You think about replication within a field and across fields differently. And this is an example from our cotton program. This is the footprint here. Every one of these dots on the map is where we have cotton trials for in 2021 for cotton. So you can see we're testing all the way from Texas up north up to, you know, north for cotton is relatively south. You know, we don't get to Canada, but you know, the Boot Heel of Missouri, and then along the coast here, and we're testing the same genetics, the inbreds, the late stage entries in every one of these locations. And early stage testing we'll do at like 6 locations that we spread out across this footprint. And the objective is to get closer and closer to that customer experience where you have the inbred, you understand that inbred performance across all those environments. And that's why if you look at plant breeding papers, there's a ton of plant breeding papers about genotype by environment interactions. There's relatively fewer papers in animal breeding about that because it's more about that overall breeding value of the bull or the cow or the sire dam combinations that you're trying to estimate. You're not estimating that genotype and trying to place it in the footprint that you're going to match for sales. The other thing where if you look at a plant breeding program, one of the things that you always see when a plant breeder talks about the structure of how they set up their program is this years of testing. And this is an example from DeKalb, the way they had their program in the 1940s, which is, it's a long time. It's like 15 years, but essentially it describes the same way that we set up a plant breeding program today, where you have a period at the beginning where you're making inbreds. Then you have years of testing where you're reducing that number of inbreds down to a different number of hybrids you're going to test. Then you make a bunch of hybrid combinations, and then you have years of testing those hybrid combinations where you're whittling that hybrid combination list down to the hybrid that's going to be planted by the farmer. And in this case, they were doing 4-way hybrids, but the same basic process is true for corn breeding program today. We do this in half this time today, so it's 7 years instead of 15 years, but that's still a pretty long time between when we make a cross till you have an inbred or a hybrid that's being sold commercially. And so this is, you know, if you look at the structure of, you know, this is a kind of a generic program where you take your cross, you're getting inbreds, you're selfing down to an F5 in this case, and then at F5, at F3, we may have some preliminary hybrid trials. F4s, you've got another set of trials, and I've tried to put here the amount of heterozygosity by this little green triangle. So you've got, we're decreasing the amount of heterozygosity until you have a clonal, essentially a cloned inbred that you're going to then be able to make reproducible hybrid combinations, and then you're going to sell pick the hybrid combinations and then sell that commercial hybrid. And why hybrids? It's even pretty easy to see visually with corn. You have inbreeding depression on the inbreds, so they're usually pretty weak looking. The hybrids yield pretty much twice what an inbred would yield, and 30% more than an open pollinated variety, and significantly more than the progenitor open pollinated varieties that they were derived from. So, this is why they're making that hybrid, this F1, then they're producing that by cutting off the tassel out of the female rows and using the male pollen from a single hybrid or an F1 hybrid in some cases with the male to make that cross, and you get a reproducible hybrid where every seed on this F1 is genetically identical, or 99% genetically identical. And that structure of your breeding program, where you're thinking about not only the value in terms of breeding value of the cross that you're making, but the progeny and how those progeny combine to get you to the final hybrid that you're going to deliver to producers, is a different way of thinking about your populations and your end goals of what you're trying to estimate. So I borrowed this from Gregor from a paper that he had where, he had this he's trying to align a plant breeding program that has this structure where you're testing a lot of, in this case, I think it's a varietal program. So you test a whole bunch of plants at F2 that are genetically distinct, and then by the time you get to fully inbred at F8 to F10, you have just a few varieties that you're testing to have the final commercial variety. Where compared to an animal breeding program where you have a selection for an improved herd that's your nucleus herd that then goes to multipliers, and those multipliers sell to producers, it's a very different way of thinking about what you're trying to estimate, because most of what you're trying to estimate and you care about in a plant breeding program is that advancement of inbreds from stage to stage, which is why plant breeding programs tended to focus on blues or mean, just the straight mean even, of that particular inbred or hybrid, because you're estimating that value of the thing and you're varying the experimental design to get it closer and closer to the production state and footprint by the time you commercialize. The other thing where you see an impact of this is recurrent selection is not as immediately valuable to a plant breeding program, because where you see your value is inbreds that go to producers. Plant breeding programs started in the 1900s as mostly recurrent selection programs that looked like an animal breeding program. They had S1 or S0 recurrent selection where they would select improvements on traits. But if your focus is getting to that final inbred faster, pedigree breeding becomes much more attractive, because your focus is not on improving your base genetics, it's improving that cloned hybrid or inbred that's going to be in your customer's hands. And you have increasing focus on genotype by environmental interaction, which is why your testing resources, as you move from early stage testing to late stage testing, gets increasing number of locations and replications as you get towards the end instead of at the beginning, because what you're really estimating is how stable is this genetics over that footprint of environments, that target population of environments. In addition, you're trying to measure the stability of that hybrid or that variety across years, because you're going to sell that clone for like 7 to 10 years mostly. So you want to have a breeding program length that's long enough that you're getting some estimate of stability across the environment as well as stability across years, which is your biggest environmental difference. And that's how you end up with a breeding program that has kind of this funnel type design, because You're making a bunch of pedigree crosses with very large populations that in the start of a program that get increasingly smaller as you get down to the few commercial that are at the end. And you can work that up and kind of get to what commercial numbers are if you think of each stage essentially as being like a 10% selection intensity. If you have a single commercial variety that you're going to commercialize and you have 6 years of testing, you end up for a single commercial variety having something like 25 or 30,000 entries that you're starting with as inbreds at the start of this funnel, because you're going to take 10% of that 25,000, you're going to have 2,500, and you're going to get 10% of that, you're going to have 250, and then you're going to go down to 25, and then 25 down to 2.5, and then you're going to select the final one that's going to go commercial. And that's the way that, like, that's where your brain is, because that's where your value is coming as from your program. So one of the shifts that is happening is what starts to align these is genomic prediction kind of changes a plant breeder's focus to where the genetic gain comes from to be more like in an animal breeding program. And maybe some of the methodology and the ways you think about population improvement start to be more and more important is part of what I'll show. So if you think of this funnel compared to an animal breeding program where your focus as an animal breeder is making sure that you understand this production system, you have breeding goals that are aligned with each of those stages of increase and with the final consumer, you have collection of information. This is also different in that, you know, especially within a breeding program, the only place you're getting phenotypes on your genetics is from your own program, whereas you could be collecting phenotypes and family relationships and genotypes even from end customers if you're an animal breeding program. Like if it's a dairy program, maybe the dairies that are producing milk are measuring the milk per cow, and they're also genotyping those cows and bulls. So you're collecting the genotypes and the phenotypes and the pedigrees of those end dairy herds to be able to help understand then the selection criteria and the breeding value, and then selecting what is the improved bull that's going to feed back into that breeding kind of nucleus herd of dairy cows, which is a different way of thinking about kind of where you gain value versus when you're generating an inbred that's going to end up in a commercial field. So this is where politics starts to like kind of play into this and interact with this, you know, and it goes back a long time. I tried to look back. I just looked at the US. There's similar history that you can find for Canada or Europe in other places in the world. You know, the end result of this UPOV is the kind of a union of plant variety protection kind of member states. So if you are a member of UPOV, you essentially have similar or equal protection on varieties, including the ability to have for protection of inbreds and hybrids. And those, you know, the green states here, countries, are the ones that are all members, and they all have very similar intellectual property laws for those. And then you can see the red states that are the red countries that are the ones that are joining or getting up to that level of protection, and yellow. And you know, there's not many places that aren't trying to protect hybrids and varieties in the same way. And if you look at where this started, you know, even into when colonization was beginning from Europe, a really important goal for those colonizing entities was plant collection and characterization and moving global agriculture around. Think about all the places that maize moved, and maize is a key part of people's diets, or tomatoes. You know, can you imagine Italian food without tomatoes and peppers and all these crops that move from the New World back into Europe, or from from China and Asia and all around, people thought of these varieties as really important and valuable things. And starting in the US in the 1790s, when they had the first Patent Act, it excluded biological innovation. So it excluded specifically protecting with a patent on things like plants, but at the same time, there was a really high emphasis from the government to collect and send back varieties of plants and maintain those. And the US Patent Office was responsible for collecting and distributing those. And up until the late 1860s, that was a very important part of that patent office's job. So I looked up some of the history of this, and in 1897, the USDA distributed 1.1 million packets of seed for free in 22 million shipments all around the United States. And those different varieties were collected and distributed and maintained by the USDA and the US Patent Office. In 1883 was when the Seed Trade Association started. They had a convention that was that was in Paris about protecting industrial property. In 1924, the Congress ended this USDA free seed program, and 2 years later, a hybrid corn co was started with hybrid, which became Pioneer, which is now Corteva. So that is not a coincidence. There were people who started to see, well, if there was a market that grew out of that change. There were a number of different increasing amount of protection that you could get as hybrids started to be sold. So there was a Plant Patent Act in 1930 that protected hybrids that were asexually produced. That would be like apples and like a lot of horticultural plants and things. Pioneer had a really key case that went in 1939 where they accused Holden Seeds, which is another seed company in Iowa, of stealing their seed, and won because their patent, they didn't have patents on the seed, but they had some protection of that IP, including that pedigree was protected. It was protected information, and that Holden's had that with illegally, and that was trade secret. That became a really key protection, and part of why pedigree breeding is so important to the modern plant breeding industry. Improved variety protection was passed in 1961. It was updated in 78 and 91. There was case law that protected then genetic engineering. And you could, starting in 1998, you could start to have even patents for hybrid corn seed that wasn't asexually produced. So all of that results in a intellectual property environment where the results of a breeding program, which take 7 to 15 years, can pay off for a company, because then they have some protection that their variety will be the genetics and the innovation that clone represents are protected for then 20 years after they start to release it commercially. So that's a big component on why there's a private, very profitable seed industry in plant breed that's supported by plant breeding. This is from the commercial business from Bayer, and you can see a bunch of different high regional brands that for corn that feed into this, and then here's Bayer's commercial brands that we sell for. So right now I work in cotton. Cotton gets commercialized mostly as Delta Pine is our kind of flagship commercial line. And why is this profitable? So this is USDA numbers for the cost of a bag of seed. So per acre seed costs over time, going back from 1976 to 2020, and you can see at 120 acres on average, $120 per acre on average in Illinois. There's millions of acres times $120 per acre. And the amount that the seed company makes on that, say, is somewhere less, probably not more than a third of that price. But, if they're making $25 or $30 per acre and selling 1 million acres of seed, that's how you end up with a company like Bayer having like $6 billion of sales a year on corn seed. So, what is the impact of this? So if you look this is from FAO. You can see this is corn yields. I'll show some cotton yields because that's my current job. That's who pays my bills. But this is where I could find a bunch of different countries. And part of why I wanted to show the different countries is you can see like increasing adoption of both a modern seed industry with the improvements that come with hybrids and modern breeding practices is part of why you see differences in some of these slopes. Some of these are because of hard environments, it is, but you can see like you did in the US for Brazil here. So that's what I wanted, one of the reasons I wanted to show this is you can see Brazil was mostly open, was a slower adoption of full hybrids. And then this is actually 4-way hybrids here up until the 1990s. And then you start to get single cross and then biotech, corn and big seed companies that are pushing modern breeding. And you get a big increase in yields that both come from adopting modern hybrids and adopting the production practices that go along with those. But that's part of why this system has been impactful. You can see this in the cotton. So this is just Delta pine cotton yields. Each of these data points represents the average of our last stage of testing. So it's the last pre-commercial testing stage that's run, and over time, and you can see we have consistent and increasing genetic gain based on investments, and these are kind of key inbreds that help move us forward, kind of jumpstart genetic gain. And that's the other thing that you see with having an inbred or a hybrid as your output of your breeding program is sometimes a single inbred or a single hybrid makes a huge difference on those data points. Sorry, that isn't this. I described this a little bit incorrectly. Each of these data points is an inbred from the year it was commercially released. This data comes from our last stage of testing, but it's not the average across the set. It's the blue for each of the inbreds that were in those trials. So, you can see kind of this jump up from a single inbred that's the best of its class that came out that year. So coming back to this hypothesis, so part of why I see a difference between both the mentality and the way we set up breeding programs and the things that plant and animal breeders care about and the business differences comes from this interaction of the biology, the business, and the politics that support it. So where do I think they start to come back together? And this was John Hickey's hypothesis, is that, on a functional level, genomic prediction starts to bring those things together. And I think that it actually is more than just genomic prediction as a statistical method, but also that the animal breeding systems, the breeding systems for animals is starting to have some of the things that look more like a plant breeding program, that you can start to replicate genetics better. You can freeze a lot of sperm from a single bull, and you can get a whole herd of cows from a single bull, or you can get the semen from a single pig and replicate that across a whole herd of pigs. And the genetics of thinking about the opportunity and also that change in that system, if there are ways that you can make clonal animal breeding programs work, I think you start to get something that starts to unify that even more. The other thing that, if that was the case for animal breeding, you might need to have an IP, like an intellectual property system, to help protect that. Otherwise, that's similar to plants. Otherwise, it may not work. It may not be profitable to do. So, this is my tagline for what the future is. And, if we've got time, we can banter about what that kind of combined future looks like. This is a corporate slide. So the tagline is from selecting the best to designing the best. But I think the combination of genomics, even more precise control of genetics through things like doubled haploid, through gene editing and transgenics, does allow us to think of kind of designing what we think is the optimal genotype, and then building that essentially, and being able to deploy that and sell that to our customers. And that's where I think being able to think innovatively on that process is where there's opportunity in the future.
Great, Brian. Thank you for the fantastic talk. It was, and especially for kind of linking it back to all the animal breeders here. Definitely helped me anyway. Definitely understand things. So I'd just like to open it to the floor. If anyone has any questions, feel free to throw up your hand and then to turn on your camera and your mic. There's Mike. Work away, Mike.
Hey, Brian. Nice to see you
Mike.
again.
Yeah.
Very, very well done talk. I think you explained that extremely well. I think you skipped over the double haploid pretty fast. I wonder if people are catching all of how important that was. Maybe you should just go back and explain how double haploids are done and why they're so important.
Yeah, let me go back to this. No, here. So this is not a double haploid process. If you can see, to make that inbred, you make a cross between your two inbreds, then you have an F1 generation, then you have an F2 generation, F3, F4, and F5. Most of the time, we're turning these generations at one per season, which tends to be, we can do 2 generations a year pretty easily if we ship seed from like the US to Mexico, for example. You can turn 2 generations a year. So if you think of this as each one of these is 6 months, this is 1 year, 2 years, 3 years, really 3.5 years till you have inbreds that you can start to increase seed of and develop commercially. Doubled haploids, there was a mutation that happened in corn that's just amazingly lucky that someone saw it and thought, that's fascinating. What happens is there's 2 different mutations. One is a maternal inducer and the other is a paternal inducer. And for some, well, we know probably the mechanism of it now, but the genome of the parent, the pollen donor, gets eliminated in the F1, and what you end up with is seed that have a normal endosperm but a haploid plant, which you can double chemically with colchicine and skip these generations of inbreeding. So you generate in a single season a fully 100% inbred line. And what you can get is then, instead of making this in 3 years, if you do 3 generations per year, you can make it in a year. If you're doing it just 2 gen a year, you're doing it in a year and a half. And then you don't have to worry about selecting or testing something that's partially inbred. You have the final inbred from the moment you start your testing program.
Yeah, this was something that I always was jealous of, that crop breeders had at their disposal to be able to create an instant inbred. And of
And
course,
not all crops have it. Like we're just starting to be able to do this in cotton effectively, right? Like we didn't have that mutation. It's really there. There was a breeder at USDA sitting in Iowa at Iowa State who found this thing in the 1930s and said, "That's really interesting," and like worked on it really for about 35 or 40 years before we were able to even make any doubled haploids. And the seed industry has benefited significantly from kind of anonymous, careful genetics. I mean, it's not anonymous. You can track down who did it and when, but I don't know his name off the top of my head. I would have to look it up, and I would bet none of the rest of you do either. And we owe them a debt.
Yeah. So that was one of the things I was very jealous about, and obviously we can't create fully inbred, animals and have that work very well. The other thing was the UPOV system, and I was wondering, back to your slide about politics. How did, to the best of your recollection, how did the plant breeding world get that kind of protection, that PVP protection, that we've so far have not been able to get that protection for elite genetics?
I think it really goes all the way back. If you think of how the governments were thinking about varieties of plants as something of value and something that was distinct, all the way back into the 1600s, I think that builds this like case essentially for how you get a system like UPOV that protects varieties and hybrids around the globe, to where we can grow in the field our varieties and have pretty good confidence that no one's going to steal that genetics or use that genetics without paying us a royalty. And like I think it traces all the way back to the way that the patent office was. Maybe they weren't allowing protection directly of varieties back into the 1790s, but they were collecting and distributing and treating each variety as a thing of value and a thing that was unique and distinct. And once you get from value, unique, and distinct to value, unique, distinct that should be protected, that's where you build a case for something like UPOV. And the other way that they made that case is that the genetics flows back, right? We may have a variety protection on an inbred, and it lasts for 20 years. We sell that inbred for about 10 years on average, but some inbreds we're selling for 20 years. The genetics then goes public after 20 years. So that was kind of the compromise that I think that was made, is there was a pretty clear understanding that that unique varieties had value, and that if they were protected, there could be a business. The other compromise there was that the business had to then give them back to be public again, right? So there is a distribution system. I can request the varieties from my competitors at Pioneer that are coming off of protection and breed with them freely. And I don't owe them a penny, but they are older, right? So I'm behind. They set up a system where there's an incentive for it to be within the system, but there's still room to get the public good. 20 years sounds like a long time to somebody who's not in the seed business, but it took 15 years to breed that variety and out on often. So, it's still often commercial when it goes off patent or off PVP.
One quick follow-up, and I'll shut up. But if you could sort of think about a counter real, an alternate reality in which UPOV didn't exist, where would plant breeding be? Where would yields be? And what would be, the cost benefit return to the farmer had that system not been there?
I think there still would have been a system. I don't know that it's as big of a business of a system. I think there would have been much more emphasis on public programs, the importance of yield and the importance of getting to that yield is still pretty important. And if you look at, the advances that were made essentially in public programs until the 1980s even, I think that system would have still existed, without something like UPOV. I don't know that you get a company like Bayer or Monsanto in the seed business without it. There are plenty of people who would say, well, that's a good thing. Like you guys shouldn't be in the business anyway. That's hurting the world. I don't know that we would have gotten the rates of genetic gain that we have. Like it would have gone up. I think we would still be on the increase because we need it. Like there's just too many damn people to not increase the amount of grain that we produce on the same acres. But I don't know that we could get as fast as we did with companies being involved. So
Thanks.
there's trade-offs there.
Thanks, Mike. Anyone else have a question? I actually have one quick question for you, Brian. It's less political, but more so just for my own understanding. And it's kind of regarding, and I think it was one of your later slides regarding, you had pretty much at the start, that one there with the breeding, and you have essentially 1 million possible combinations that you can trial and you can run. And then you end up with 1 to 3 final products.
Yeah, let me find
that are actually commercialized. So my kind of question is around, like, once you get to those finalized populations, is for your next, when you start off again, essentially at the bottom of the funnel, what is the impact on like the genetic diversity that you have within that next cycle? Are you reincorporating everything that you had initially to again try and find new combinations and stuff like that? Or what is the thought process?
I kind of flew over that, but if you notice, there's these 2 other arrows here, right? Mm-hmm. This green arrow is where we're picking parents, and we can pick parents from any of the stages essentially, right? So we could pick from within this genomic prediction bucket even, we predict something to be awesome. It could be used as a parent immediately, or you could wait until it went all the way to the commercial. There's a trade-off there. You have high confidence in anything that's made it into this commercial bucket, but it took a lot longer to get there, right? It took us 7 years to determine that it was good enough to go commercial. There's probably other things here that you can have good enough confidence in that you could pick that's earlier, and you're managing diversity on 2 levels: diversity within the breeding program and diversity within the commercial output, right? And you can manage the commercial output being lower diversity because you're going to turn over hybrids. So if you have a hybrid that's weak in some way, that's got a disease resistance, or that's not good enough, for example, you can replace that with a new hybrid if you've got the diversity in your starting bucket. So as a breeding company, we think a lot about that diversity that we have at the beginning. We care about the diversity that we have at the end because we want to make sure we have what our customer needs. But in order to support the customer of the future, right? There's the diversity the customer needs now and diversity the customer is going to need 10 years from now. The 10 years from now bucket is, diversity is still in that top bucket. And so that's where we really put our focus on making sure we have lots of genetic diversity, is at the top of the funnel.
For sure. Thank you. Any last, I see we're over time, but if anyone has any last quick questions, I am not seeing any.
I'll end with my legal disclaimer slide. So Mike Lohais, who used to be the IP boss, doesn't report back that I did a bad job. I did show the cautionary statements of forward-looking legal disclaimer side. All opinions are the express opinion of Brian Gardunia and not a Bayer Crop Science. No, perfect. But I really appreciate the chance to talk to you guys. I really do think there's lots of opportunities right now to think innovatively with genetics, both in animal and plant breeding programs. And it's a fun time to work in this field where there's lots of things that we know about the genome. There's lots that we don't know. And I think that the things that we're going to learn and be able to do in animal and plant breeding, and the challenges that we have to figure out how to solve are really important. So thanks for the chance to come.
Great. Thank you, Brian. Really appreciate it. And thank you everyone for joining today. We'll see you all again next week.